Your extractable study came back clean, every compound identified, all levels below concern. Your dream is relieved, the hard part is done. Here is what most people don't realize and what most concern here is acceptables tell you what could migrate from your container closure systems, as on the other side leachables tell you what actually does migrate into your product on yourself under your storage conditions. Those two lists don't always match, and the gap between them is where patients get hurt and submissions get delayed. Hello, I am Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we help pharma and biotech companies design and execute acceptables and leachables programs that hold up to regulatory scrutiny. Today we are going to from lab bench to market the full leachable study workflow stage by stage, including the parts that most teams underestimate until they are already behind. Before we move forward, if you have found anything valuable in this channel, please like and subscribe. This gesture of appreciation means really a lot to us. Let me show you where programs typically run into trouble. A team completes their acceptable study, call it month 6. They have a profile, they are confident their packaging is understood. What they often don't account for is that the leachable study, the in-use simulation with the actual drug product under actual ICH storage conditions, can run another 12 to 18 months before you have the data you need. And the analytical evaluation threshold, the AET, which defines what you have obligated to characterize, gets set too loosely far too often. Teams who start the leachables program late almost always find something at hour 19 that they wish they had found at month 6. So what does a leachable study actually look like? You're exposing the real drug product to the real container closure system in the real formulation under accelerated, intermediate, and real-time stability conditions. The product itself becomes the extraction medium. Accelerated conditions at 40 degree and 75% relative humidity simulate long-term self-aging in a matter of weeks. You test upright and inverted orientations to maximize contact between the product and the stopper, the valve, the tube, whatever the material of concern is. You pull samples at defined time points, typically 0, 1, 3, 6, 9, and 12 months. And every time point goes through the full analytical battery. And the analytical battery is where the lab work gets interesting. No single instrument sees everything. GCMS, usually with headspace or SPME extractions, captures your volatile organics, solvents, processing residues, low molecular weight antioxidant fragments. LC-MS gets your semi-volatile or non-volatile compounds, the antioxidants themselves, UV stabilizers, plasticizer, degradation products. ICP-MS handles elemental impurities, which is critical for parenteral and ophthalmic products under ICH-Q3D. And when something shows up in your sample that does not match anything in your extractable library, an unknown, you move to high resolution mass spectrometry and sometimes NMR for structural characterization. That unknown identification workflow is where programs routinely lose 3 to 6 months they didn't budget. So let's talk about the single most important number in your entire program, the AET, the Analytical Evaluation Threshold. The Analytical Evaluation Threshold defines the minimum level at which any leachable, known or unknown, must be reported, identified, and assessed. It's calculated from the safety concern level, your route-specific tolerable daily intake, and a safety factor that typically runs 10 to 100 times. Get the AET too high and you miss compounds that may matter to the regulator. Get it too low and you have obligated yourself to structurally characterize every trace impurity in a complex biological matrix. Most ENL deficiency letters that we see trace back to one root cause, and that is an AET that was set without a documented defensible justification. When a compound shows up at or above AET, the question becomes, what do you do with it? This is where the threshold cascade begins. Above the identification threshold, you need a structural ID. You have to know what it is. Above the qualification threshold, you need a full toxicological risk assessment, knowing what it isn't enough. At the safety concern level, you have a regulatory problem that requires action, not just documentation. The TTC framework and Kramer classification give you a defensible starting point for qualification when compound-specific toxicological data doesn't exist. But for high-risk dosage forms, parenteral inhalation, pediatric use, regulators increasingly push for compound-specific data even when TTC defaults would otherwise apply. 3 cases illustrate what happens when the analytical work runs behind the regulatory conversation. First, Aprex epoetin alpha, where a formulation change combined with a switch to an uncoated rubber stopper generated an immunogenic leachable. Over 277 patients developed pure red cell aplasia. The outcome was a global formulation overhaul. Second, MDI valve elastomers, a compound from a valve O-ring, exceeded the safety concern level at accelerated conditions, forcing an 18-month program delay and a complete change of valve supplier. And third, biologics in pre-filled syringes, silicone oil from barrel siliconization drove protein aggregation in multiple monoclonal antibody programs, generating BLA hold responses and pushing the industry toward low silicon and silicon-free container system. At Resolve Mass, we have supported teams through investigations like this, where a single uncharacterized peak in a chromatogram become a 12-month program to the cases took different on surface. The failure underneath is always the same. The analytical work wasn't finished before the regulatory conversation started. Now, here's the 5-stage workflow that prevents all of that. Stage 1, study design. Set your AET, select your time points, define your analytical method suite, establish your acceptance criteria. This is where 80% of your downstream workload is either managed or created. Stage 2, controlled extraction. Confirm your contact material inventory and build extractables library you'll use to attribute peaks in your stage 3. Do not skip these steams that move directly to an in-use simulation without a solid extractable libraries. Routinely find unattributed peaks they cannot explain to the regulator. Stage 3, the leachable study itself. in-use simulation running in parallel with your stability program. Stage 4, analytical characterization, identification, quantification, unknown structure elucidation, and stage 5, safety qualification, TTC assessment, Kramer classification, SEL comparison, toxicological risk assessment, and the submission document. The teams that invest in stage 1 rarely face emergencies in stage 4. So, what should you walk away with this session? Number 1, a leachable study is a separate product specific program. It is not a byproduct of your executables works. Number 2, the AT must be calculated and documented, not assumed. Number 3, unknown compounds above the identification threshold need structural characterization. Budget 3 to 6 months if you find them. And 4, start this program at IND, not at NDA. The timeline will find you either way. If you are working on an IND, an ANDA, or a biologics program, and if you need a CRO or CDMO partner who work with you as a partner, Resolve Mass Laboratory supports teams from first study design through submission ready packages. Thank you so much for watching this video till the end. Before you go, I want you to do 3 quick things. Number 1, subscribe this channel right now, because the more you subscribe, the more valuable pharma insights and expert tips and industry updates we can bring to you. Also, please comment and let us know what did we do wrong and what did we do right, because the more you give us the feedback, the better we will be able to make videos for you. And number 3, share this video to at least one person who would find it useful. You never know who is stuck on exactly this kind of technical challenges right now. I will see you in the next time. Until then, goodbye for now.